All right. Well, so welcome everybody. It's really my pleasure to introduce Anders Håkansson. So Anders did his PhD at Lund and discovered HAMLET, which is a topic of which he'll talk about today. So I won't say anything more about it. Then he went to do his a postdoctoral fellowship with David Briles, University of Alabama, learning investigating the pathogenesis of Streptococcus pneumoniae, and then did a second postdoc with Mike Wessels here in Boston, and then took a faculty position at University of Buffalo. And there, two things. One is he developed, and this is when I first met him, when he came and visited, and I think Andy was his host, and talked about the interaction of the bacterium with influenza virus, and described some really interesting studies about signaling from mammalian cells into biofilm localized bacteria, and had developed a mouse model of co-infection with influenza and Streptococcus. And it was so interesting to me, in fact, that Vesma Joma in my lab, an immunology graduate student, actually took that model and tweaked it so that we could see disease. And so he and I collaborated on that project, and he was absolutely instrumental in us being able to get that model up and going. The other thing I will mention is that he moved back to Sweden to take his current position. I think he's your director of, hold on, I got it here, division chief of experimental medicine in the Department of Translational Medicine at Lund University. And of course, that was a loss for University of Buffalo, but those of you who know Elsa Bulganem, who is a postdoctoral fellow in my lab, also working on strep, actually took the position that he left. So at least she was happy that he left. So anyway, so we've been collaborating, and it's a real pleasure to have him both as an affiliate member of CIMAR, and gives us an international stature, I suppose. And also he's doing some really interesting work, and I'm hoping that people not only find it interesting, but he's quite interactive. And if there's things that people want to talk to him about, I'm sure he's open to thinking about stuff. So welcome, Anders.
Thank you so much for the really nice introduction. So yes, I will focus today on HAMLET primarily, even though I do a lot of host pathogen interaction work as well, as John was saying. So the topic today is to talk about this HAMLET protein, and I'll give you a little bit of background, and I'll talk about how it kills bacteria and how it can be potentially used to reverse antibiotic resistance. So I know that let's see here if I can get this going. Good. Yeah, so antimicrobial resistance, of course, this is something that is not this is something that is very familiar to this audience, and you probably know more about that than I do. But it's what's kind of put on the map, I think, in 2014 with the WHO kind of coming out saying that we're heading in a post-antibiotic era here if we don't do anything. And a couple of years later, they added on a priority pathogen list with 12 pathogens besides TB, which was already a major problem, where you need urgent kind of therapeutic and preventive strategies to combat these increasing resistant organisms. And data that has been coming out lately from the European network of antimicrobial resistance, for example, looking at data from 2007 to 15, they showed that at 2015, they had about 670,000 infections with AMR pathogens and about 33,000 deaths, which is kind of an increase with 250%. The CDC, of course, came with its threats, antibiotic-resistant threats analysis or report in 2019, and showing that you have about 2.8 million infections, 35,000 deaths with AMR pathogens. It seems like the trend might be kind of slowing down a little bit compared to 2013. And then more lately, we've had kind of the United Nations came out with their report estimating about 700,000 deaths from AMR pathogens that may be projected to be 10 million or so in 2050. And just the other day, I think it came out in PubMed 2 days ago, the antimicrobial resistant collaborators came out with the global burden, an estimate, a global estimate showing that probably it's not necessarily 700,000 deaths, but more like 1.3 million deaths that are directly attributable to kind of AMR pathogens, and about 5 million deaths that are associated with these types of infections. So it's a huge problem, as we all know. So the organisms I will talk about today is primarily Strep pneumoniae, where we have a lot of penicillin resistance, which is a serious threat according to the CDC. About 30% strains are resistant to one or more antibiotics. I will also talk a little bit about Mycobacterial tuberculosis, which of course is a major problem with multi-drug and a couple of totally resistant kind of strains as well. And I will talk about Staph aureus to some extent, where of course methicillin resistance is a big problem. Vancomycin resistance is on the increase, and there's the new antibiotics. We start seeing antibiotic resistance against those as well. So that's the background. So what can we do then? Well, so in this series already, we've heard some very interesting things about, for example, using current scaffolds to make new antibiotics with the scaffolds that we already have. Andy was talking about phage therapy, which is kind of very promising as well. We have biologicals coming with monoclonal antibodies, again, specifically directed against antibiotics, against pathogens. We can, of course, also, not enough vaccines against these pathogens, of course, as well. Immunomodulators and prebiotics or probiotics could be helpful to improve our immune responses and combat antibiotics-resistant bacteria. And then I'll talk a little bit about sensitizers as well. So we already on the market have beta-lactamase inhibitors that can kind of resensitize bacteria that are resistant to penicillin. There's also quorum sensing inhibitors that can kind of stop biofilm formation, that is, and sensitize biofilms to antibiotics. People have been working for a long time with antibiotic efflux pump inhibitors, and that is kind of progressing as well. So what I will talk to you about today is HAMLET, which I guess will kind of fit in two of these categories. One would be that it's a directly bactericidal agent, but more interestingly, for an application purpose, I think would be that it might be a sensitizer to sensitize resistant bacteria to the antibiotics they're resistant to. So to give you a little bit of a background, these are the people that have been doing most of the work. So I haven't done all of this work myself, of course. Lots of people have been involved. And the discovery from the beginning, so I discovered this when I was a PhD student. So this is essentially my kind of, in a way, my thesis work on a slide. So what we ended up doing was that we were working with breastfeeding at the time, knowing that breastfeeding is protective against infection. So we started fractionating the milk in different fractions, and one of those fractions ended up being really interesting. So we looked here at binding of, in this case, Strep pneumoniae to epithelial cells that we scraped from our nasopharynxes. And when we added this fraction 6, that we called it at the time, that we didn't know what it contained, we could see that all the bacteria went away. So the bacteria couldn't bind anymore. However, when we used a cell line, in this case, A549 cells, but we used about 40 different types of cancer cells after that, we found that not only did the bacteria not bind anymore, they actually disappeared, but the cells looked really funny as well, which they didn't do when we used healthy cells. So it ended up that these cells were dead. So this fraction killed the cells. And when we looked at the distribution of HAMLET, here we're using labeled HAMLET, we could see that it binds to cells, and it actually binds to all cells, but it gets inside of tumor cells, but not into healthy cells. And the mechanism for that is actually not clear yet. The pattern here that you see in the cytoplasm ended up being a co-localization with mitochondria. And what you can see on the next one here is if we then look at the polarization of mitochondria, the polarization goes away. So you get depolarization in the presence of HAMLET, of the mitochondrial membrane. And that activates caspases, and it activates a whole cascade of apoptotic signaling that leads to DNA fragmentation and all of the events that we usually connect with apoptosis. So here we have like a high molecular weight DNA fragmentation. So that was kind of how everything started. And this fraction 6 ended up containing what we call HAMLET. So it's a protein-lipid complex from human milk. It consists of alpha-lactalbumin, which is the most predominant protein in human milk, not in other milk sometimes. So there's beta-lactoglobulin in bovine milk that is more predominant, but there's lots of alpha-lactalbumin in human milk, about 2 grams per liter. So in its native form, it produces lactose in the mammary gland, in the Golgi apparatus. And for reasons that people haven't understood for a very long time, it's secreted in these enormous amounts into the milk, where it doesn't produce lactose. So it's doing that in the mammary gland cells. So when the calcium ion that is binding here to the native state is coming off the protein, which could happen in the gastrointestinal tract, in the gut of the child, then the protein structure opens up slightly. It becomes in different conformation. And oleic acid, which is quite predominant in human milk, but not in most other mammalian milks, can bind to this protein in that conformation and make a stable complex that is then active against cancer cells. So native alpha-lactalbumin has no anti-tumor activity, and whereas the HAMLET form has. And the reason we call it HAMLET, it stands for human alpha-lactalbumin made lethal to tumor cells, which is one of my colleagues came up with the name. We thought it was appropriate because, well, HAMLET is about death, so we thought that was good. And also the castle where this takes place is a Danish prince, and the castle is this is where I'm located right now in Malmo and at Lund University. And it's kind of across the water over north of Copenhagen here. So we thought, well, that could be a pretty good name. It's localized to where we are, and it's about death. So we called it HAMLET. So what about the bacteria? So as I showed you in the slide before, the bacteria also seem to disappear when we did this. So it kills bacteria, it kills tumor cells by attacking mitochondria. And mitochondria or bacteria are kind of predecessors of mitochondria. So maybe there are some kind of, does it kill bacteria? And are the pathways in that case potentially similar in that case? So that was a question I had towards the end of my PhD. I started working on that then and have continued with it since. So yes, it kills a number of different bacteria. Here are 2 examples. It kills Strep pneumo. It kills Mycobacteria tuberculosis. If we look at the Strep pneumo data, we can see here, the red line shows you a dose-dependent killing with HAMLET, starting at about 10 to the 9 bacteria. Alpha-lactalbumin, the native form of the protein, shown here in black, has no bactericidal activity. And oleic acid does have bactericidal activity, but the concentration found in the complex doesn't have the same activity as the complex itself. So the complex itself seems to be good at either delivering the oleic acid or doing something combined in the conformation it has. If we look at Mycobacteria tuberculosis as an example, we see direct killing, that the amount of bacteria in the presence of in this case, about 300 microgram per ml, which is, I think, 18 micromolar. You see complete death of the bacteria. And we can also actually, if we load THP macrophages with mycobacteria, and then add HAMLET afterwards, we can actually kill the intracellular bacteria as well, which is it's really nice. We've also found that other streptococci, like this is erythromycin-resistant, Strep pyogenes, or clindamycin-resistant Streptococcus acalacti, or group B strep, both are killed, as you can see down here. And the MIC levels, this is growth curves showing MICs, are approximately around 20 micrograms per ml, or 50, a little bit higher for GBS. So there's a number of bacteria that are being killed. If we look at Strep pneumo and looking at the mechanism then, and try to compare what is happening, we can find that, yes, if we do purified mitochondria, using green labeled HAMLET and red labeled mitochondria using MitoTracker. We can find a co-localization of HAMLET together with the mitochondria. And we see the same binding in green here of HAMLET to strep pneumo. And interestingly, we use MitoTracker here to stain the bacteria as well, it works really well. We can also see depolarization in both mitochondria, which I showed you before, and in the bacteria. So depolarization of the membrane of strep pneumo is happening at approximately the same rate and at the same concentration. And we also see other features that are somewhat apoptosis-like, you could say. So in tumor cells, what you tend to see is that the nucleus becomes condensed and fragmented. And if you look at the nucleoid in strep pneumo, we can see that this is untreated cells. And when they get treated with HAMLET, we tend to get this very strange structure of condensation of the DNA in the bacteria. And in both tumor cells in strep pneumo, we see this DNA fragmentation, at least on the high molecular weight, and there's very similar between the tumor cells and the bacteria, suggesting that there is at least some similarities between the types of deaths. So if we look generally, we then have tried a number of different bacteria, and HAMLET doesn't kill all bacteria. So strep pneumo, as I showed you, mycobacteria, certain streptococci, as well as Haemophilus influenza, which tends to be a gram-negative in that case, all of those are being killed, whereas other gram-positive and gram-negative bacteria like E. coli, Pseudomonas, Staph aureus, and so on are not being killed directly by HAMLET. So this indicates that for some reason there's some kind of specific activity against respiratory tract pathogens. It also seems like it kills bacteria resistant to various antibiotics. So we've tested a number of different strains of strep pneumo resistant to a variety of different antibiotics, and they're all being killed by HAMLET. So it seems to be a different mechanism. We've also tried to make HAMLET resistant clones, which have failed at this point, which I guess is a positive thing. We tried to do that to find a mechanism. But we have tried really hard and we haven't succeeded so far. So when we started out this part of this, we suggested that this could be a novel mechanism that we wanted to explore to potentially develop agents with less risk for resistance development. So now I will show you the mechanistic stuff that we've done so far using strep pneumo as the model organism. So we started out looking at trying to identify potential HAMLET targets in strep pneumo. So we used biotinylated HAMLET and tried to pull down from a lysate of pneumococci whatever HAMLET was binding to, using streptavidin beads. And we used a cross-linking to be able to capture as many proteins as possible. So we compare here the HAMLET pull down, which is beads alone, which of course also pull down unspecific proteins as well. So the specific proteins that we got, that we only found in the HAMLET pull downs, and then the ones that we'll talk about today are the ones in red. So we found that components of the hydrogen ATPase seem to be binding to HAMLET. We were interested in ion transporters because we know that ion transport, and especially calcium transport, was important for death of tumor cells, but we couldn't find anything that looks like that, but we still investigated. We found a serine-threonine kinase that we have looked into a little bit, and we found that two major glycolytic proteins, the fructose bisphosphate aldolase and GAPTH, bound HAMLET really well, and we couldn't see that anywhere else. There are, of course, a number of other things that we haven't reached, haven't looked at yet, and those are found here that could be involved as well. So I'll show you the data with the ones in red. So, of course, the depolarization that we see, so here to show depolarization that is dose-dependent, increasing over time, could be an association with ion transport as well as the proton motive force. So this is my little picture of what the hydrogen ATPase might look like. So we have a barrel here that where the transport is occurring, and then you have an ATPase on the inside, the F1 portion of this, using ATP to kind of, in this case, for these bacteria to kind of expel hydrogen out so that the pH on the inside is higher than that on the outside. So these bacteria don't have oxidative phosphorylation, they don't have a TCA cycle, so they use this mechanism to kind of regulate their pH on the inside rather than making ATP. So the way we looked at pH here was to load the bacteria with BCECF, which is a fluorescent dye that measures, that kind of reacts with hydrogen ions. So we loaded the bacteria with that. We then equilibrated them. So the pH on the inside is somewhere in close to 7.6. And then at time point A, we added in CCCP, which is a proton ionophore. And then, of course, we get kind of equilibration. We also added in optogene. So optogene is used to kind of, it's an antibiotic that actually binds in the barrel of the F0 portion of the ATPase. And it's usually used to kind of differentiate strep pneumo from other virulent streptococci that are resistant to optogene. So we used optogene as a positive control here, and we can see that this also kind of decreases the pH, suggesting that the ATPase is kind of inhibited. And in red, you can see that HAMLET does the same thing. So we get an inhibition of the hydrogen ATPase that kind of equilibrates the pH to that of the outside. So you have some kind of role of this ATPase. We also looked at a number of other types of transport mechanisms and other ions, and the ones that we found results with were sodium and calcium. So again, if I show you the depolarization profile here with HAMLET alone in red, if we now add in, for example, a calcium transport inhibitor, ruthenium red, which is usually used as a mitochondrial calcium uniporter inhibitor, but has other functions as well, we see a reduction in the depolarization. And if we use amiloride, which is a sodium transport inhibitor, we almost get complete reduction of or inhibition of this effect, of HAMLET's effect. So we could be talking about sodium transport and calcium transport separately. There's also a possibility that we have a sodium-calcium exchange mechanism going on. However, there are no transporters like this that we can identify. There are no, based on the eukaryotic kind of sequences, there are nothing that looks like any of these transporters in the pneumococcal genome or any other genomes that we have found. So we don't really know what this is. But we looked at kind of calcium influx to begin with. So as we add in HAMLET, we can find that using either calcium 45 or using TRF2, we can see a rapid influx of calcium into the cells. And we can also see that when we now add these inhibitors, if we add the calcium transport inhibitor, we get a reduction of about 50% of the calcium influx. And if, interestingly, if we add the sodium transport inhibitors, either amiloride or an amiloride derivative that is more specific to calcium-sodium exchangers called dichlorobenzamyl, both of those completely blocks the calcium influx. So there's a connection between the sodium and the calcium transport. To see that that was actually involved in HAMLET-induced death, we used them to look at viability of the bacteria. So here we just have the buffer control. We have about a little bit over 10 to the 8 bacteria. If we add HAMLET alone, we get a reduction of about 3 logs. And if we add ruthenium red, amiloride, or the dichlorobenzamyl, the sodium transport inhibitors, we almost completely rescue the bacteria from HAMLET-induced death. So the dichlorobenzamyl kind of indicates a little bit that we have a sodium-calcium exchange kind of mechanism potentially, or that they're at least linked in some kind of way. So to test that a little bit further, we ended up making an experiment where we used a PBS-based buffer, but we also made a PBS-based buffer where we reduced the amount of sodium, adding in gluconate to kind of keep the osmolarity up to 25% of the original PBS. And when we did that, we saw that at the lower concentration of HAMLET, we get about 2 logs of death in the regular PBS, but we get about 6 logs of death if we have a lower amount of sodium, suggesting that because sodium has to work against its gradient, the concentration of sodium on the outside is much higher, whereas calcium, of course, goes with this gradient. So for this exchanger in that case to work, having lower sodium on the outside will facilitate the effect, and that's kind of what we see here. So we think that we have some kind of sodium-calcium exchange mechanism, but we have no idea what this is, and we haven't found any molecule that looks like any type of sodium or calcium transporters so far. So that's what we're working on. When it comes to serine kinase activity, that has ended up being quite interesting as well. So there is a very well-characterized serine kinase in Strep pneumo called STICP. I think it was isolated and identified in 2004. And it's an important protein involved in phosphorylating a number of different target proteins. It's involved in cell division and a number of different things in the cells. And it has one transmembrane domain and has the serine kinase domain on the inside and has 4 PASTA domains on the outside, which is penicillin binding protein associated with serine kinase activity. So that's not the protein that we found. What we did find was an open reading frame that ended up, when we looked in InterProScan, to also have a serine kinase domain. The original paper on STICP actually showed that when they mutated STICP, they still saw a lot of serine kinase activity. So about half of it disappeared, but there seemed to be another protein that they didn't identify. So we think that this could potentially be that. It looks a little bit different. It has more transmembrane domains. It has a very similar kind of serine kinase domain, but on the outside we have a xanthionine synthetase domain instead. So that's what it looks like. So to begin with, we just wanted to look at does HAMLET actually induce phosphorylation at all? So this is using phospho-threonine antibody. And what we find is that on the first 3 lanes shows you HAMLET alone, 2 different concentrations. So there's a reduction of the phosphorylation of 1 or several bands around 45 kilodalton. And at 100 kilodalton, there's an increase in the phosphorylation of some component here. So there's a modulation of the phosphorylation where some proteins are phosphorylated, others are dephosphorylated when HAMLET is added. And then adding in staurosporin to these two in the presence of HAMLET, we get essentially a reduction of phosphorylation generally, which we would expect that staurosporin would do. We then got a mutant of STICP, and we made a mutant with this, what we call it STICX so far. We don't really know what to call it yet. So STICX seemed to be involved, whereas STICP did not seem to be involved in HAMLET-induced death. So this shows the opposite. So this is not the viability, this is the amount of death that you see when HAMLET is added. So in the absence of inhibitor, we get about 7 logs of death. And then we add in staurosporin to D39, regular pneumococci, and we get a reduction of death down to between 1 and 2 logs. And the same pattern is seen with the STICP mutant. We get, again, about 7 to 8 logs of death, which is reduced by staurosporin down to 1, 2 logs of death. But when we mutate STICX, this new protein or new open reading frame, we find that now we have a reduction in death. It's still not completely eradicated, but we get about 3 logs of death, and staurosporin doesn't have an effect on that amount of death. So we think that staurosporin is acting on the STICX protein instead. So we haven't proven yet that we're trying to kind of express it. It's a little tricky as a membrane protein to express well. We're trying to express the domains. It's been a little bit tricky. So we haven't proven that it actually is a serine kinase yet. And that's what we're working on. We are also doing kind of other types of studies with this protein. But it seems like it's involved in HAMLET-induced death in some way. And the final thing that we talked about was that we found that fructose bisphosphate aldolase and GAPDH were 2 proteins that came out and kind of binds to HAMLET. So this is an overlay on the lysate, and we find 2 distinct bands here that it binds to. If we then make a recombinant protein and run those out and do an overlay, we see that HAMLET binds really well. Interestingly, though, alpha-lactalbumin, the native protein, also binds actually pretty well to both the proteins. And this is just the streptavidin background, just a little bit of background binding. So important to know for what of course what glycolysis is doing is to give you pyruvate, and primarily in glucose-based media we get mostly homolactate fermentation. That's the only thing that we end up getting. You could get mixed fermentation as well, and on the way you get production of ATP, of course. So those are the endpoints that we have measured here, and this is the main source of energy for these bacteria. So the first thing we did was to look at these enzymes. So we had them recombinantly expressed, and we could look at activity of both of the enzymes. And what we found was going from high concentration here to lower concentration is that both alpha-lactalbumin and HAMLET inhibited both FBPA and GAPDH pretty well. And alpha-lactalbumin, of course, doesn't have any bactericidal activity, so this was a little bit surprising. But what we ended up seeing was that if we did this in vivo instead, only HAMLET has inhibitory activity. So this experiment is set up in the way that we take the bacteria whole, and then we add HAMLET, alpha-lactalbumin, or the oleic acid component of the HAMLET complex for 10 minutes. And then in the cold, we wash the bacteria, we make it lysate, and then we measure the activity. And the activity here is measured by the decrease in NADH based on FBPA activity. So what you can see here is that now alpha-lactalbumin works perfectly fine, similar to the D39 control. But if we add a sublethal concentration or lethal concentration of HAMLET, we get a reduction or complete inhibition of the FBPA inside of the cells. So it seems like HAMLET can reach its target from the outside and get inside of through the membrane, just like what we saw for tumor cells. And this could be also seen if we looked at lactate production from the cells or ATP production. As soon as we add in here at 5 minutes or 10 minutes HAMLET, we can see that at that point, the lactate and the ATP production just stops, but we have no effect of alpha-lactalbumin if we add that in at 5 minutes. And also we can see that if we are kind of energizing the cells in different ways, if we add a lot of glucose, the blue line here, to ramp up the glycolysis, we can find that we get a lot of, it becomes much more difficult for HAMLET to kill the bacteria. But if we inhibit glycolysis with 2-deoxyglucose, which is a version of glucose that cannot be metabolized, and inhibits glycolysis, then we get a better effect of HAMLET. So it seems like modulating glycolysis seems to be important as well. So this takes me to kind of the somewhat complex model of HAMLET-induced death that we have at the moment. And there's lots of pieces of the puzzle that we don't have. So HAMLET blocks the hydrogen ATPase. It also seems to induce kind of a calcium influx that is sodium dependent. It also induces some serine kinase that we think we have a new one that we found. And it also blocks 2 of the enzymes involved in glycolysis. So how all of this is orchestrated, if this is happening sequentially, if this is happening in parallel, or how they are connected is what we're working at with at the moment. It is interesting to kind of note at least that, of course, inhibiting glycolysis will decrease ATP production. And ATP is required for the hydrogen ATPase and the serine kinase to work, so there is at least a link there. It is also known that the serine kinase can phosphorylate proteins, including FBPA has been shown to be phosphorylated by serine kinase activity that is not STIC-P and could potentially be this STIC-X protein. But this is still unknown. And then, of course, how this cell death is executed is also not clear. Are there proteases and DNases involved, as we can see in apoptosis in eukaryotic cells or not? We don't know. Similarly, the link between, so depolarization is occurring, of course, of the membrane. We can see that. How much of that is the protein most in force and how much of that is other ion transport happening? We don't necessarily know. We also don't know how much calcium is involved. It's known that if you look at MAP kinases or other serotonin kinases in eukaryotic cells, they're often calcium activated. So calcium might have a role here. There must be a link. So there's a lot of things that we need to do, but at least we have a starting point, and we have a number of different targets that could be interesting to look at in the future as targets for antimicrobial agents other than HAMLET itself. So future here, of course, the green ones are the ones that we have some kind of idea of what the molecules are, and that they seem to be targets for HAMLET. The sodium and calcium ion transport mechanism, we don't know what it is. We haven't found it. We don't know what molecule it is. Similarly, all of these other things like proteases, DNases, regulators, and transporters, we haven't got into that. We need to continue. So we hope that kind of also additional proteome and transcriptome analysis might help in identifying these pathways in a better way, and potentially together with you, like Andy and John, TNSeq or even CRISPR libraries might be helpful in kind of identifying more the pathway of how HAMLET works. So the next thing, of course, we wanted to do was, okay, can we use HAMLET now to treat infection in some kind of way? So the first thing we did was to actually do the hardest thing you can do, which is to try to reduce pneumococcal colonization. So pneumococcal colonization is, of course, done through biofilms, and biofilms are really resistant to antimicrobial agents. So it might not have been the smartest thing to do. And what we saw was, yes, HAMLET doesn't really have a good effect. We get a slight reduction, but it's just quite minor. So when we did this, and we're pursuing kind of other types of assays at the moment with pneumonia and other types of models. But this is kind of how we started out, and we didn't really see a good effect of HAMLET alone. So then we thought maybe if we combine it, we do combination treatment with antibiotics, we would see some kind of effect. So this is something that Laura Marks worked with starting in Buffalo, and we'll continue doing a lot of this work. So what she ended up doing was, this is of course, HAMLET is, D39 pneumococci is of course kind of sensitive to HAMLET at some concentration. So if we go down and use a sublethal concentration and do an MIC assay, so this is a growth curve, the bacteria grow up and then they stay in stationary phase, and pneumococci have an autolysine, so at some point they will lyse and die. So this is kind of the growth curve that we see. So HAMLET alone at the sublethal concentration or subcidal concentration did not do anything, and penicillin at the low concentration did not affect the growth. But if you combine the two, we got a really good effect and got down to an MIC. So we can see that here in terms of fold reduction of the MIC. So if we take the D39 strain that is already sensitive to both penicillin and HAMLET, we can still get a 5-fold reduction of the MIC by combining the two. If we take a penicillin-resistant strain, on the other hand, we can get an even better effect. We get approximately 20-fold reduction of the MIC. Similarly, if you take erythromycin, D39 is sensitive to erythromycin, we can get about a 3-fold reduction. But if we take an erythromycin-resistant strain, we get a 300-fold reduction. We can actually, it becomes sensitive to erythromycin again. Gentamicin we've added in here. We don't have a gentamicin resistant strain in our library here, so we're just taking 2 strains and we get about 4-fold, which is similar to what we get for the other antibiotics if they're already sensitive. So this was kind of interesting that combination treatment here could actually make them much more sensitive to the antibiotic they're resistant to. So we then thought, okay, biofilms are really hard to do something about. So this shows you an SEM picture of a biofilm of pneumococci in quite low resolution. So this is the kind of complex network of the bacteria makes. So if we now again take D39, we add penicillin at a high concentration, 100 microgram per ml, we get about 2 logs of death of the biofilm. HAMLET at a high concentration, a little bit less than 2 logs, but if we combine them, we get up to 5 logs of death. And the effect is even better if we take the penicillin-resistant strain. So the dotted line here is the total biomass. And of course, penicillin doesn't do anything. HAMLET itself doesn't do much, but if we combine the two, we can almost completely eradicate the biofilm. So then we thought that would be, now we're getting somewhere. So now maybe we can go and do something with colonization. So colonization being forming a biofilm, maybe a combination here would work much better. And it ended up that we did. So if we take a sensitive strain again, we can find that if we add in this case, gentamicin, we colonize the bacteria, the mice for 48 hours. And then we take out the tissue, the nasal tissue, and we see how much bacteria we have per tissue. And then we increase the concentration of gentamicin in the presence and absence of HAMLET. So HAMLET, as I showed you before, didn't really do much by itself. But the combination treatment here actually works really well. So the red dots here shows that even at 3 microgram per ml, we get a large reduction kind of in the bacterial load in the nasopharynx. And at 100 micrograms, we have almost complete eradication of the bacteria. So it's much better with combination treatment, even for an antibiotic sensitive strain. And when we talk about the resistant strain, the colonization rate here, the burden is a little bit lower. The strain doesn't colonize as well. But of course, the black dots here shows you that if we add increase in concentration of penicillin, we don't really see much happening because it's a resistant strain. But if we combine this with HAMLET, we can again see an almost complete eradication of the bacteria. So combination treatment here ended up working really well. But what about the HAMLET insensitive organisms? What about these organisms that didn't where HAMLET didn't have a direct killing effect? Well, the interesting thing is we have both gram-positive and gram-negative organisms here. So we did an experiment with depolarization of the membrane. And interestingly, we found just like for strep pneumo, we get a depolarization event happening when we add HAMLET at time 0. And if we use E. coli, which is not being killed by the bacteria, or Staph aureus, which is not being killed, we also see a depolarization event happening after adding HAMLET. And as I told you, only in the case of strep pneumo do we end up getting bacterial death. For the other ones, that doesn't happen. So the question was then, is this enough, this depolarization event and whatever other things are happening to the insensitive strains, is that enough for them to be sensitized to antibiotics? So this shows you an methicillin-resistant Staphylococcus aureus strain, the NRS 384 strain. So this is again an MIC assay. We grow up the bacteria. These are the untreated ones. If we grow them in the presence of a quite high concentration of HAMLET, this is 100 microgram per ml, which is 6 micromolar. Nothing happens. They grow equally well, or potentially even better. If we add in 1 microgram per ml of methicillin, they grow equally well. But if we combine the HAMLET and the methicillin, then they don't grow at all. So we've done this on a number of different strains and for a couple of different antibiotics. And what we can see that all the resistant strains, so all the first strains except for the last one, all of those are resistant to methicillin. We get a fold reduction that is somewhere between 8 and 32 times the original MIC. So that's the kind of reduction we can see when we add in HAMLET to the system. We of course get a less reduction when the strain is already sensitive. We can also see this for erythromycin. So an erythromycin resistant MRSA strain, we get about a 4-fold reduction in the MIC. For gentamicin, we saw somewhere between 16 and 160-fold. Unfortunately, we don't have as good of an effect with vancomycin. It's about a 2-fold reduction, but at least we see something. So for a number of different antibiotics and for a number of different strains, we can see this happening. So again, we thought maybe we can at least give it a go and see if we can do something with biofilms and in vivo colonization. So when we look at biofilm eradication, we can see, yes, again, we get a good effect. So HAMLET doesn't have any activity at all on these biofilms. And as you can see here, for the resistant strain, 250 microgram per ml of methicillin doesn't do anything. But in combination with HAMLET, we get approximately 3 to 4 logs reduction in the biofilm biomass. With a methicillin sensitive strain, we get a little bit of a better effect with methicillin, about 1 log of death, but together with HAMLET, that increases to over 4 logs of death. And this is something that we need to optimize a little bit. The colonization rate here with this strain that we used was very low, about 10 to 3 organisms per tissue. But we can still see kind of at least a trend, and it ended up being significant. But if we look at methicillin alone, there's of course no difference in the colonization rate. But in combination with HAMLET, we see a reduction, not complete eradication, but we see a reduction in kind of the bacterial load. So that's very promising. Similarly, we've seen that Mycobacterial tuberculosis, at least in vitro, is also can be sensitized. So at sublethal doses of HAMLET, we can get kind of a better effect of various drugs, and we've tested a number of drugs. So we can sensitize mycobacteria as well, having HAMLET present at the same time, which comes to the conclusion. So what we've done so far, if we look at the sensitizing activity, looking at different antimicrobial agents, at this point, we tested these groups, you could say. So we have like beta-lactams or cell wall synthesis generally, and we try kind of antibiotics from these groups as well. And it seems to be working with a number of different groups of antibiotics. So erythromycin seems to be it works very potently with that. It works very well with penicillins and so on. So those are things that we move forward with. When it comes to kind of for example, the WHO priority pathogens, I already talked about mycobacteria, I talked about Staphylococcus, strep pneumo. But we've tested, at least in vitro, we tested all of these ones, and it seems to be working. So mycobacteria, yes, it works. We've done some work with Acinetobacter baumannii, with Enterobacteriaceae like E. coli and Klebsiella, seems to be working there. Enterococcus as well. And Haemophilus influenza, I showed we had a direct effect, and we also have a sensitizing effect. So this is also work that is kind of we're expanding on, and that is coming along. So it seems to be promising that it has a pretty broad kind of activity against both the gram-positive and gram-negative organisms when it comes to sensitization. So with that, I just have some acknowledgements. The funding that I've had for this over the years, as well as the people here in Lund. The people in red are the ones that have been involved, and I showed you them on the picture before. In Buffalo, these three were the main people involved in the work. David Browse, of course, have provided me with a number of strains, and we've done some work with Melinda Pettigrew at Yale associated with this as well. So with that, thank you so much for your time and for listening. And if you have any questions, please, I'll be happy to take them.
Is my audio working now?
Yes.
Oh, great. Okay. Yeah. Great. So thank you very much, Anders. So so questions. So can I start off with a question?
Oh, yes, absolutely.
So it seems like, just thinking about the mechanism of action, it's pretty unusual. Obviously, it interacts with these multiple proteins and seems to inhibit them. And is it in some ways acting the same in some fashion? In other words, if you take lactalbumin without the oleic acid, does it interact with only I know it interacts with some. Does it interact with all, or is there something different about? Is there some specificity, I guess, in the targets, in the binding of its targets?
Yeah, so it's a kind of it's a really good point, because so when we do an alpha-lactalbumin pull down, for example, some of the proteins, like you saw, the GAPDH, the glycolytic enzymes, and some of the quite prevalent enzymes are being pulled down. The hydrogen ATPase is not being pulled down. Those proteins are not. Those seems to be HAMLET specific. And the serine kinase seems to be HAMLET specific as well. So some of them, there is a stickiness, general stickiness, that they are pulled down with alpha-lactalbumin as well. And for some of them, there's not. So it's not kind of generally one way or the other.
So I guess, yeah, I'm getting at the question of whether or not its interactions, it has this broad specificity of substrates. Is it using the same domain, for example, to interact with all of these substrates? Are the same?
Yeah, so that's a good question that I don't have the answer to. We do have a version. It's been a little tricky. There is a version we feel that we need potentially need the oleic acid to have the activity. So we have a version where we at least have half of the protein. So half of the domain can be made recombinantly and will bind the lipid and also has kind of very similar activities. So that's kind of the closest to kind of what they're saying, is it this part or is it that part that is doing it? So we've been doing some work with that, but we haven't tested all of the targets with that yet. So I can't necessarily say exactly what the mechanism would be and which domain is kind of involved. But we have some tools, but not really good tools to kind of look at it.
So let's see. So Emmy has a question in the chat. Can you tell us more about tumor cell killing and whether you've evaluated for mammalian toxicity?
Yes, so that has been done. So we have tested, so when it comes to tumor cells, it seems to be a very broad activity. It's about, like I said, around 40 different types of tumors that we have tested. We've also, of course, tested healthy tissue, and it's been done in animals and it's been done actually in humans as well. So we've done some human studies, not by me, but my former mentor, Katharina Svanborg, has been involved in doing those, so that we don't have any kind of side effects in terms of toxicity on human cells, healthy human cells. So it seems to be very tumor specific. And why that is still not clear. It seems to have something to do with being able for the protein to be able to enter into the cells, which it can't for the healthy cells. And only tumor cells seems to be taking it inside, or it gets through the membrane in some kind of way. So that is unclear.
And then in macrophages, like it can kill intracellular bacteria and macrophages.
Yes.
It presumably is getting into macrophages.
Yeah, so it's kind of what we found there was that it actually follows, comes into the phagosome. So it kind of follows the bacteria through phagocytosis and ends up in the phagolysosome.
Okay, great. Maya Nadampali asks, are the concentrations of HAMLET you've been evaluating in your assays comparable to what's actually found in human breast milk?
Yes, so that's a very good question. So when it comes to alpha-lactalbumin, we have 2 grams per liter in the milk. And what I have been using here is, so that would be 2 milligrams per ml. And we've been using somewhere about 20 times less is what we're using in our assays than what you would find when it comes to native alpha-lactalbumin milk. It's really hard to we don't have tools to say how much of the protein in the milk that ends up when it goes through the gut to actually become HAMLET. That we can't really measure. So what percentage of the alpha-lactalbumin actually ends up being HAMLET, I don't know, but if it's 1/20th, if it's 5% of the total amount, then we're in the range of what we're using in our assays.
Marcia Osborne asks, do you know if HAMLET works by itself in vivo if you pre-treat with it rather than trying to eradicate a biofilm?
Okay, so sorry, one more time. The question was,
do you know if HAMLET works by itself in vivo if you pre-treat with it rather than trying to eradicate a biofilm? And maybe, Marcia, if you want to unmute, you can elaborate.
Yeah, I think I understand what, so if you were to, oh, sorry.
No, that's okay. Go ahead.
No, so if you pre-treat, if you kind of have like kind of more of a preventive treatment. So if you try to kind of use it either at the same time as you add the bacteria during colonization rather than a treatment, we haven't actually done those experiments. So I don't know. I know if you try to make biofilms, for example, we don't need very high concentrations of HAMLET to block biofilm formation. So if it's present there all the time, they can't actually form biofilms. But we haven't done that in a mouse model. But it's possible that could work really well, or better than trying to treat the biofilm after it has been formed.
Right.
And that would be kind of probably what breastfeeding is doing. So when you're breastfeeding, you have continuously being fed alpha-lactalbumin through, and that kind of flows over your nasopharynx all the time. So that could be the reason why you have a lower kind of colonization rate or lower risk of getting big biofilms and so on in the nasopharynx.
Andrew Plout has a question.
Yes.
Yes, I wanted to ask you two quick questions. One is, oleic acid is a monounsaturated acid, as far as I can recall. Are there any other fatty acids that are even close to it that can actually be suitable for binding to lactalbumin?
Yes. So the thing seems to be it's interesting because alpha lactalbumin doesn't seem to be the specific aspect of this. You can take bovine alpha lactalbumin and you can make a complex, and people have been working with that, and they call it BAMLET instead of HAMLET. So that seems to work if you mix an oleic acid with bovine alpha lactalbumin, and we've tested a number of other alpha lactalbumins as well. So that seems to work pretty well. But oleic acid and linoleic acid as well, which is the version that has two double bonds. Both of those kind of bind and form a complex that is active. And oleic acid is the most, I think 40% of the fatty acid content in human milk is oleic acid, and then linoleic acid is a close second. It seems like we need a C18, so both of those are C18. It has to be that long, but exactly where the double bond is, it has to be a cis bond. Can't be a trans bond, but a cis bond will work, and two cis bonds will work as well. So a number of fatty acids will work. And it's just interesting that the ones that make up the majority of fatty acids in human milk, or primate milk, we should say, are the ones that seems to be working in this complex.
Thank you. I want to ask one other quick thing. I noticed in one of your early slides that there was a time dependence on how long you expose this to pneumococci. What's going on during that period of time? 5 to 10, 15, 20 minutes. It gets at John's question of, is there a common mechanism, almost catalytic in appearance? What's going on?
You're absolutely, exactly. So it seems like depolarization is something that starts within the first minute or so. So we start seeing depolarization really quickly. Other effects might take a little bit longer. So like the hydrogenated PAs, but it could be a little bit depending on the assay, how reactive the assay is as well. So that takes a little bit longer. It's a little bit slower of a process, even though it starts quite immediately. We haven't measured kind of serotonin kinase activity itself because we don't know. So it's much harder to kind of measure that. So we don't know exactly when that would start. And when it comes to glycolysis, that's also quite immediate. So all of these things start happening within the first minute or so. But exactly the stoichiometry of which one comes first and so on, has been really difficult to tease out.
Good, thank you.
Okay.
And I guess, Andy Camilli had to leave, but he gets the last question, and he left it in the chat. Is HAMLET toxic when delivered IV? If so, will it be restricted to mucosal therapies?
It probably will be restricted to mucosal therapies for a different reason, and that is that it's inactivated in serum. Mm-hmm. So serum albumin seems to also bind the oleic acid equally hard and make you get a complex between HAMLET and albumin, which inactivates a large portion of the activity. So this is going to be a very and that's why I'm looking for targets, because this is going to be a difficult thing to administer intravenously because activity goes away. So rather than being toxic, it's the other way around.
Well, great. This was really interesting. And I know some of us are going to be meeting with you tomorrow. But thanks a lot. Thanks for joining CIMAR, and thanks for giving this talk.
Well, thank you for inviting me. It was a pleasure.
Okay. All right.
Thank you so much. Have a good day.
You too.